So this is a phase 3 trial testing a new compound called Custirsen together with chemotherapy used for second-line metastatic prostate cancer that has resisted already to not only hormonal therapy but also to first-line Docetaxel chemotherapy. So as probably you will know, prostate cancer is a very frequent disease. It's being treated mostly by hormonal therapy, the first-line treatment when we need systemic treatment. And hormonal treatment works very nicely in almost all men who suffer from this disease, but it doesn't necessarily work very long. After 1 or 2 years, perhaps sometimes longer, the disease tends to resist, and until recently we didn't have many options to propose to these patients. The very good news from the last 5 years is that a series of compounds that could be shown to increase overall survival in this situation of what we call castration-resistant prostate cancer. 2 chemotherapy agents, Docetaxel and Cabazitaxel, 2 androgen receptor axis targeting drug, abiraterone and enzalutamide, and 1 bone targeting agent, radium, and this is because metastatic prostate cancer is a bone disease. Now, having said that, none of this compound is able to achieve a cure in patients with metastatic disease. So we still need more treatment, and we need treatment that can reverse or prevent the resistance to these drugs. Custirsen that is shown here is a cytoprotective protein. So every time a cell, a cancer cell, will be attacked by either hormonal therapy or chemotherapy, Custirsen will be expressed. And it will try to protect the cells against the harming from the treatments we're using. It pretty much gives the time for the cells to survive, if you will, by various means, and I won't go into too much detail, but it really prevents the apoptosis that we are trying to achieve with hormonal treatment or, again, chemotherapy. So Custirsen, which is shown here, which is the drug I'm going to talk about, is an oligonucleotide antisense that is really preventing the making of a protein. So by combining chemotherapy plus Custirsen, the goal is really to achieve a better job than chemotherapy alone. So the rationale was based on multiple experiments in the last 15 years in vitro and in animal models, mostly conducted in Vancouver by Marty Gleaves' group, suggesting a strong biological effect of Custirsen and an inhibitory effect of Custirsen. Then we moved to clinical development with Custirsen, and a randomized phase 2 trial was conducted and reported some years ago, suggesting that indeed combining Custirsen together with chemotherapy can achieve a better job with approximately a 40% reduction in the risk of death in this randomized phase 2 trial, again in metastatic castration-resistant prostate cancer. So we designed the AFFINITY Phase 3 trial as a large Phase 3 trial in the second-line setting, so in very advanced patients. They had all failed not only hormonal therapy but also chemotherapy with first-line Docetaxel, and it was a quite straightforward randomization of a standard of care, Cabazitaxel prednisone, plus or minus Custirsen, which is an IV drug that we give on a weekly fashion after a loading dose. The patients continue treatment until progression, cancer progression, toxicity or completion of 10 cycles, and the primary endpoint was survival. This is the main results of the trial, and unfortunately, by intent to treat analysis, there was no superiority of Custirsen when added to chemotherapy. We looked at different subgroups. Perhaps it was a little more active in what we call poor prognosis patients, but still no overall survival benefit. Having said that, we've tried to look at what happened to serum Custirsen, and was that because Custirsen finally is not a good enough target, or was that because the drug is not good enough to target Custirsen? And actually, in the control arm, you don't see much effect whether Custirsen is being lowered or not by chemotherapy. Now, in the experimental arm, it's another story. as you can see here, and those patients who achieve a marked decline in cholesterol while on Custirsen do much better as compared to those who do not achieve this effect. So again, declining cholesterol seems to have an effect. But again, perhaps the drug is simply not potent enough to have this biological effect being translated into overall survival improvement. So in conclusion, addition to Custirsen to second-line chemotherapy with Cabazitaxel did not significantly improve overall survival. There was no new safety signal, and generally speaking, this is very well tolerated as an add-on to chemo. No imbalance of active drugs beyond progression can explain the lack of overall survival benefit. We looked at that because we have now, again, many drugs that can postpone death, and it was well balanced between the two arms. So again, two possible questions or answers. Either cholesterol is not relevant enough as a target in this disease, or, and perhaps more likely, the target was not inhibited enough with the compound we have at the moment. The second and last conclusion that I have is really about the pre-requirement that we need for the future when designing a new phase III trial. Because in this trial, we had a strong biological rationale. We also had, I think, a strong clinical signal. Again, overall survival was improved or apparently improved in randomized phase II, but it didn't really select all the patients based on biomarker. There was no molecular certification, and I think this is what we need to achieve now for future development in prostate cancer. Thank you very much.